I'm going to pause for Zoom recording. Okay, and so
stop your share.
Yeah, great. Okay, so Warren, you should be able to.
Yep. Right. Thank you very much, Nikki and Annabelle, for giving me the opportunity to sit here in my office and present all the way to across the North and maybe the South Island. It's just a shame I haven't been able to come over and visit and do this in person for the last 2 years, but let's see what the second half of this year offers us. And I think you might see us visiting once or twice if possible. So my talk today is broken up into 2 segments. One is just an introductory talk. So welcome to everybody who's a newbie and has heard of Nanopore, knows a little bit, but doesn't really have a full comprehension of the technology and what it does. And also welcome if you're using it. And this is an update, and hopefully my update presentation and Mike's detailed tips and tricks will be of use to you. And then I think Nikki and Annabelle are thinking of maybe some bioinformatics workshop-y sort of thing later in the year to fill the depth of information out that was just too much to do all in one sitting. So further ado, I will get on to my presentation. So Nanopore has a rather unique and lofty goal that drives the direction of our technology development, and that is to enable the analysis of anything by anyone, anywhere. And that's a pretty big goal to have if you want to cover research, health, industry, education, biosecurity, food security. These are all areas that are actually adopting Nanopore now, and it's becoming wider spread internationally for this whole range of applications. New Zealand especially has embraced Nanopore technology across a wider range of applications than I've seen in any other country, which is really exciting. This is a snapshot demonstrating the diversity of on-site sequencing that's possible with a small portable sequencer that needs minimum additional hardware to run an assay, needs very small compute, additional power, etc. So we've done sequencing on the space station, on beaches, in submarines, in the snow, a huge variety of locations to show you what's possible. Rainforests and caves and other places, deep, dark, wet places, you can still sequence with Nanopore. So why would you consider Nanopore sequencing? So that's a good question. Well, we offer a wide range of really unique benefits that you don't get with any other technology. For a start, it's a direct RNA or DNA sequencing without PCR amplification and those problems. There's no intermediary fluorescent reporters for nucleotide identification, etc., which provides other challenges to technologies. It's direct analysis of the sequence from the genomic sample. We're not just a short read technology. In fact, we're going to rebrand ourselves. We're read length agnostic. You can sequence down to 20 base pairs all the way up to over 4 megabases. Whatever is in a library that you load into a flow cell can be sequenced. And another feature of our platform is real-time analysis. So while the sequencing is occurring, it's actually generating the nucleotide and modification data at the same time on the sequencer. It's very flexible. You can start, you can stop runs, and real-time enables certain applications like adaptive sampling that I'll be mentioning more in some later slides. So there's some really exciting applications that can come with this real-time analysis. In terms of the data we can provide, it's the widest of any genomics platform. And I have confidence in saying that because I've represented probably about 15 genomics technologies over the last 20 years, and none of them compare with what we can help with. Having that read length range, the genomics direct, and being able to analyze modifications is just does my head in. We can help with assembly, chromatin and epigenetic analysis, gene fusions and splice variants and gene expression. It can be done at the single cell or it can be done in bulk samples. And structurally, we can look at long structural variants, small indels and SNPs, and these can all be phased across huge segments of the genome. We can provide a whole genome analysis or several methods for targeted analysis. And again, whole transcriptome and metagenomics are two key applications. So how does it actually work? This is a schematic, a cartoon of a sequencing in progress. So we embed a bacterial membrane, bacterial pore in a synthetic membrane. We apply a charge difference across the membrane, and that provides the electromotive force to pull the DNA or RNA across the membrane. As it's flowing through the pore, there is a flow of ions from one side of the membrane to the other, and that flow is disrupted by the nucleotides as they're going through the pore. And that disruption is measured with some bespoke microelectronics that generates a squiggle plot that we then deconvolute with algorithms to identify ATCG and the base modifications. The second component of the system is the motor protein that you can see in purple attached here to the end of the RNA or DNA, which is really the only library preparation step that's essential. So it's very simple technology to set up a library run. Now that motor protein has three functions. It has helicase activity to unwind the DNA or RNA, uses ATP to do this. It helps locate the strand at the mouth of the pore, and it acts as a handbrake to slow the strand down. It slows it down to about 200 to 400 base pairs a second. And if it didn't do this, the strand would go through the pore at millions of bases a second, and we'd have no time to measure anything. So it's all a very well-controlled sequencing environment. And those pores have then been arrayed into flow cells that have a range of scale. It's no good if you want to enable the sequencing of anything by anyone, anywhere, if you've got one instrument that's one scale and one size. You really need a continuum, and we have that available now. At the smallest end of the scale is the flongle. It has 126 channels. It'll generate a 0.5 to 1 gig of data on average, and the price is under $90 per flow cell. And then you've got the three systems in the middle that run MinION flow cells: the MinION Mark 1B, the Mark 1C, and the MinION Mark 1. The MinION Mark 1B, many of you will recognize because it was released probably 7, maybe 8 years ago, in our very early release days. And that's our core basic sequencer that runs the MinION flow cell, USB connected into a computer for base calling, and you're up and running. The MinION flow cell will generate 10 to 30 gigabases of data. It has a maximum potential of 50 gigabases in a perfect world, but we've got customers that have generated well into the 40s and on a routine basis. So it's getting right up there. So the three systems, the other two systems, the Mark 1C and the Mark 1, have built-in GPUs. The Mark 1C is a single flow cell system with a built-in GPU for base calling and a touch screen for system control. And the GridION has a 5-position deck for 5 flow cells and has a powerful GPU for real-time base calling of those during the run. And then the big boy of the group is the PromethION, 24, 48, and 2 that I'll be mentioning in the next talk. The flow cells from the PromethION have 3,000 channels, and these can generate 100 to 150 GB of data routinely. We've got customers that have generated over 200 gigabases. It does depend on the sample, the library prep, and a few variables, but certainly well over 100 gigabases is routinely achievable. Now, our company has some core values that make us really different from every other genomics company that I know of. And this is built into our DNA. Sorry for the fingers there, but it's built into our DNA. We want our technology to be accessible to everybody. And to do that, we make it available with no capital cost. And I can explain that further later. Pricing is transparent. It's on our website. It's done in 4 currencies worldwide. Unfortunately, US dollars and Australian dollars are not there, but we work in US dollars for simplicity. The pricing is the same for every customer everywhere in the world. You could be a little lab in Invercargill or the Broad Institute, and you pay the same price for the same product. There's no special mates rates for institutes or for countries. We've got a very level playing field on pricing, which I particularly like. Upgrades are included because we're a continually evolving technology. If you commit to using our platform, we commit to providing the upgrades and an upgrade path, whether it be consumables, software or hardware. We're continually improving the hardware, for example, gridirons and PromethIONs. As more powerful GPUs become available at a lower price point that we can bring them into the systems, we do incorporate them and you get free upgraded hardware. You don't have to buy $1 million box every 3 years just because they make improvements in it. We're very different to those other companies. And community feedback is incredibly important to us because it's how we do our product development, which is iterative, not linear. Most companies spend a couple of years developing a new product or part of their technology, and they release it when it's 100% capable and locked down. We don't do that. We actually develop a new part of our technology till it's useful. It's not fully developed. It'll have bugs. It won't do everything that it might be capable of eventually, but we release it early release on our website, so you're aware of this. And we value customer feedback on what it does well, what it doesn't do well. And this is folded into the next round of product development. In fact, we've got a bulletin board on our community site where researchers post the features they want to see in our products. And these, I know for a fact, feed into product development meetings and product management meetings, and they discuss which of those they're going to tackle first. So it really is, it's not an empty statement that we value community feedback. So as I stated, we provide direct PCR-free DNA sequencing with no conversion steps in between, so we can detect base modifications. There's, I think, a dozen or so DNA-based modifications that are known, but on the right-hand side here, you can see RNA mods from a publication in 2017. I think there's over 170 modifications there. Our technology is capable of measuring these, hopefully all of them eventually, because they will have either biological consequence or they will affect the biology in some way and will be important to measure. I'll touch on this a little further, but it's definitely a work in progress, but it's the only technology that can easily look at this range of base modifications. The one that's probably of the most importance at the moment is CpG methylation. I'm just going to showcase quickly a benchmarking study we did about 1.5 years ago. We took 2 ENCODE data sets from a public repository, bisulfite sequencing 50-fold coverage, and we did our own 20 times coverage with Nanopore. In this graph, you can see the very tight distribution across the whole genome from the Nanopore data. There was very little of the genome that got less than 20-fold coverage and very little of the genome that got more than 20-fold coverage. So you've got a very even coverage, which is important. With bisulfite sequencing, a 50-fold coverage data set actually has a lot of the genome well under 50-fold coverage, and it has some of the genome well over 50-fold coverage. So you're not going to get accurate analysis across the whole genome compared with the Nanopore data set. And there's a couple of reasons for that. One of them is our platform has no GC bias. It doesn't matter whether it's high or low, it doesn't affect the bases going through the pore and the detection of those bases. Because we have longer DNA that we sequence, it just maps better. Therefore, you need lower depth of sequencing to give the same analysis, to do the same mappability. So we give a lot more of a data set is mappable to the genome, and that means we can identify more CPGs in that sample. So in this example, bisulfite sequencing, it identified 80 to 85% of known CPGs in that human genome sample that was used. With nanopore sequencing at 20-fold coverage, that's less than half the coverage of bisulfite, we can identify over 90% of the CPG sites. And at 40-fold coverage, almost, I guess it's probably 97, 98% of those are identified, which means you just get more data, more meaningful data from your sample. And using one of our, I guess, best-in-class modification callers that's about to be replaced by a better one. And you can see here in the bottom of the graph, the bottom of the diagram here, where you align the reads and we actually call the individual bases as being modified or not modified. And then you can aggregate these into a stack plot to identify the degree of methylation at those different sites and how they align with the genome browser. We also align very, very closely. So it's all one question is to identify the CPG sites, and then how well do you correlate with bisulfite sequencing for the detection of methylation at that site? Because there's no standalone standard that you can compare against. So comparing with bisulfite sequencing, very high level of correlation, 0.933. In fact, it's higher than that these days. This is a little bit of an older slide. And we also compare very well with methylation arrays. This is a screenshot from a customer in Queensland who did her own internal comparison with the EPIC arrays, and she confirmed that our correlation is over 0.9. So you can have a lot of confidence in moving from arrays and bisulfite sequencing to nanopore sequencing with methylation analysis, that you're going to be able to identify all of the sites that you've had previously and add additional content to those. Just touching on the ability to read any length of DNA from 200 base pairs, from 20 base pairs up to over 1 megabase. Until recently, we had a filter on our system which limited the low end to 200 base pairs. You can now go down to 20 base pairs, which is important for some applications like liquid biopsy and cell-free DNA. In fact, there are several groups in New Zealand who are now using this ability for a short read length analysis of native DNA in the blood for additional as a step forward from short read sequencing, which won't give you methylation of those segments either. Then you can look at FFPE analysis. 1 to 2 kb is perfect for full-length isoform analysis. If you want to look at repeat regions, centromeres, telomeres, you really need 100 kb or larger for spanning those. And for genome assembly, adding a little bit of 1 megabase data really improves the contigs. So we provide that huge range of applications across the board. And that long read is really important. It's fine if you've got E. coli or Lambda. You don't really need more than tens of kbs to sequence their genomes. But when you're looking at tulips or lungfish, which have 34 and 40 GB genomes, you're not going to get it without ultra long reads. Plants are similar. They're challenging in their own right, highly repetitive genomes. As most people are aware, plants replicate their whole genome historically, and they're multiploidy, and therefore you get modifications of these secondary, tertiary, complete genomes, and they've got a lot of structural repetitive regions. A nightmare for short read data. You really can't do it with properly. You can have a go, but it's not going to be great. These are some comments from recent publications. Gapless and telomere to telomere assembly of plants is now possible. It's a game changer with these ultra long reads, looking at TEs in common plant genomes, that's transposable elements. And we can also deliver 5-methyl-C and 5-hydroxymethyl-C analysis of those sequences, which hasn't really been easy to do in the past. Looking at comparing with short reads, as most of you will be aware, with short read sequencing, you really need to collapse the sequence you generate to get sufficient overlap. It's very difficult to look at individual alleles, whereas with the complete long strands, we don't need to collapse that data set and you get allele-specific resolution. And this is really important in looking at, for example, human diseases, repeat expansions, which are involved in a lot of neurological diseases. We can span entire repeat regions in a single read to provide a much better level of analysis. And we can give you the methylation around that as well. Real-time analysis has a very practical, as a practical aspect. Nikki and Annabelle are running a gridiron in their facility there. And because of the nature of our platform, you can start and stop a run whenever you want. You don't have to run it for like 72 hours and come back and see what you've got. You actually monitor the run during the run. And if you've got enough data, you can stop it. You can put another flow cell in a different location and start it at the same time. All of these locations can run simultaneously, but they're all independent. And this can be really useful in time-critical applications, for example, in leukemia, where in a particular study, they were looking at a BCR-ABL fusion transcript, and they got 100 reads, over 100 reads in 15 minutes. So they just stopped the run, wash the flow cell, put it in the fridge, and start analysing your data. Another application to take advantage of our real-time base calling is adaptive sampling that I mentioned earlier. And this is worth going into a little detail because Nikki and Annabelle are getting this application up and running on their gridiron. So it's a method of targeted sequencing that involves no manipulation of your DNA or your library. Hard to believe, I know. It's done bioinformatically. So what you do is you prepare your library whole genome as you would normally. The difference is you identify the regions that are of interest to you. You identify them with their nucleotide sequence, typically around 200 base pairs, 200 to 400 base pairs, and some flanking region as well. And you identify all of your targets and you download this database into the sequencer. So what happens is you start the run and the pores start sequencing. Within the first half to 1 second, they're able to identify the first 200 to 400 bases, and they do a comparison with the database that you've downloaded. If those sequences are on that database, that's fine. Nothing happens. It just keeps sequencing and that whole strand gets sequenced. If, however, that sequence is not on that database, the system controls each pore individually, and we reverse the polarity of that pore. That strand is ejected, it's reversed again, and it can continue sequencing from the library above. What this means is you generate a data set of your targets, full-length targets, and a whole lot of 200 to 400 base pair off targets. You end up with a much smaller data set, and you end up with enrichment for the targets that you're interested in. And this has 3 general applications. As I said, enrichment of what you're wanting to look at. But we can also do depletion. If you've got a microbiome sample and you don't want to sequence the human components, you don't. And we can also balance the sequencing, which is going to be especially important in clinical applications where you've got a range of targets and you have to reach a minimum depth threshold to have a quantitative analysis. So you can keep sequencing the ones you want until you've got enough of them accumulated, and then you can go offline and analyze that data. As I said earlier, our library prep is very flexible. It's really simple. This is a snapshot of pretty much all the library prep kits we have available. But the first 2 there, the ligation and the rapid field kit, are the most commonly used. And all they are is attaching our motor protein adapter to the end of the DNA. You can do it with ligation, and there's a nick repair step and a bead purification step in this particular workflow, and that provides the very highest quality library prep and therefore data set sample sequencing for that run. But we also have a field or rapid kit, and these use a transposome complex to both cleave the DNA and attach the adapters. They're a very simple 3-step shake and bake and sequence in 10-15 minutes, perfect for in-field use. Because there's no nick repair or bead cleanup step, you don't get quite as much data out of the run, or I should say some of the data is filtered, but it's incredibly valuable and it can be used in most applications as well. And then we've got PCR amplification kits, because sometimes you'll just want 5 or 10 kb of known sequence. You want to attach barcodes so you can multiplex samples, and the methylation isn't important for that particular study. Imagine looking at Parkinson's disease and you've got a region that you just want to cover for a large number of patients, and that's very targeted, that application. Perfect for that. RNA, I'm going to touch on in a little bit more detail in a slide in a minute. And then we've got a couple of methods of targeted sequencing as well as adaptive sampling for achieving that. So transcript and transcriptome analysis is, I guess, fully enabled within Nanopore in that we enable the sequencing of complete RNA fragments with all of our kits. And so you get full splice information that can then be looked at for differential gene expression or isoform expression. It's strand specific, and we can do things like fusion protein detection. These are the kits and some details about the kits for RNA preparation. We've got the first kit here is our direct RNA sequencing kit. So that just takes genomic RNA that you might have purified with triazole. We need 500 nanograms of poly A, so it is rather a lot, I understand that. And the motor protein for this assay runs at a slower speed to DNA, and therefore the total output from a flow cell is lower than the direct DNA sequencing. But the beauty of this kit is there is no enzymatic processing of the RNA at all. No reverse transcription, no PCR, no anything. So you're just measuring exactly what's in that sample. And this can be ideal for validation studies, for example, because it's orthogonal to other methods. And then we've got 2 kits, a direct cDNA sequencing kit, which does give you a first strand cDNA synthesis. And then it feeds into our DNA sequencing kit. It needs less sample, 100 nanograms of poly A. And because it's using our faster motor protein, you get more sequence generated, 10 to 30 million reads, depending on which flow cell. Then there's the PCR cDNA sequencing kit, which has that first strand synthesis, and then 13 to 15 rounds of PCR amplification. And this enables you to work with really small samples, such as single cell or sample limiting neurological samples, environmental samples, etc. So it has its place in the scheme of applications. And I'm going to be talking about this kit a little bit more in my next talk, because we've got a new version just been released. The direct RNA kit, as I mentioned previously, does identify the base modifications because they're not stripped away by PCR or reverse transcription. However, if you look at the workflow, you will see there is an RT step in there, and we do that to help stabilize the native RNA. That strand is not sequenced at all. It doesn't have the motor protein attached to it. Only the native strand does, and this assay works incredibly well. And as an abrupt end to my talk, I'll see you at the next presentation. Over to you, Nikki.
Thank you. I'm just trying to find.
You can see the end coming there.
I know, it's great. Let me share my screen real fast. Yeah, so thank you so much, Warren. That was really interesting, and it gave a good overview of all the different applications that nanopore sequencing provides. Yeah, so basically I'm just going to give a little bit of a brief overview of what we do here at Auckland Genomics, and then we're going to talk a little bit about, Annabelle's going to speak to some of the projects that we have that we've been doing with nanopore sequencing. We have our new gridiron. She's coming back in. Yeah, so I think a lot of people on this call, I can look at some of the names and I can see a lot of people are at the University of Auckland. So if you're familiar with the campus, we are right on the corner of Wellesley and Simon Street. We're on the 7th floor of the Science Center. We have this beautiful new lab space that's just great. We have a very knowledgeable team, I like to say. So we have Jaehoon Wu, who's part of our next generation sequencing team, Christine Boxon, who does all of our Sanger sequencing, Annabelle Wibley, who's here with me right now, myself, and then Professor Ant Poole is our sort of academic lead. He helps with strategy and things like that. So here's our contact details as well. And I'm going to just go through this quickly. So I just wanted to sort of highlight some of the stuff that we do in case you didn't know. Like I said, we have a great lab space, lots of stuff. A lot of what we do obviously is sequencing, next generation sequencing using Illumina. So we have 3 MySeq machines, and we have partnerships with several different groups that enable us to do NovaSeq, all different levels of Illumina sequencing. So we can do pretty much any level of data delivery for Illumina sequencing, even though I know we're talking about Nanopore today, but we consider ourselves a national platform. So we obviously service the researchers here at the University of Auckland. A lot of people, about 30% of our University of Auckland user groups are from the Faculty of Science, but we also have Faculty of Medical Health Sciences. We have people from engineering. We have someone in the waiting room. Okay. We also, though, a lot of people I think aren't familiar with the fact that we service a huge range of external users as well. So you don't have to be with the University of Auckland to use our services. So almost half of our revenue that comes in or work that we do is for other universities, Crown Research Institutes, and we have some Australian universities and occasionally some commercial services as well. Yeah, so we have Sanger sequencing, so single read, sort of old school, but still a lot of people use this for genotyping and just single sequences. Christine Boxon runs this for us. So it's at a relatively low price point per sample, and she runs it twice a week, so you can get your data back pretty quickly. She always likes to say she gives free troubleshooting advice as well, which we do too, to be honest. We also offer microarray services. So more and more actually people are using this. Other places are sort of closing theirs down. So we're getting a lot of people who are still using microarrays, and that can be a very good alternative if you don't want to do RNA-seq. Depending on how many samples you do, it can be cheaper. And there are certain applications where people are still interested to use microarrays. They're very easy, and the analysis is quite simple, to be honest. Many people, I think this is such a really neat aspect of Auckland Genomics, is that we have a huge range of bookable equipment. So we purchase and service and maintain lots of very expensive and great equipment that many different people can use. So we have digital droplet PCR, which is sort of shown here. We have a 384 well qPCR Quant Studio. We have a whole range of PCR machines, obviously, that anyone can use. Bioanalyzer, NanoDrop, Blue Pippin for isolating and selecting DNA at different sizes. We have qubit, and we can do 96 well plate or even 384 well plate qubit reading. We have 2 liquid handling robots. We have EPMotion and a biomech robot. Obviously, we have mid-ion nanopore sequencers that people can book out and take with them if they want to go in the field or use it in their class. So yeah, some of these machines obviously need a bit of training, so we offer training so that you get set up and you know how to use it. And some of them are fairly simple, so don't need much training. Okay, we also, really quickly, we offer single cell sequencing using the 10x Genomics platform. So we have the Chromium controller in our lab, and that allows, you can take a population of cells and basically emulsify them in an oil emulsion, and then barcode and get the entire transcriptome of a single cell. And so you can do that for up to 80,000 cells. And so that's something that a lot of people are interested in. And you can, typically people do that with Illumina sequencing, but Nanopore has just come out with the ability to also, to do that with Nanopore sequencing. So you can get full length transcripts. That's quite new though, to be honest. So yeah, we're getting there. We collaborate with the flow cytometry group at Auckland, at University of Auckland. So Anna Brooks shown here, so she can, if you have a population of cells that you're interested in, she can sort them and then we can sequence them for either 10x or other applications. We've been doing a bit of single cell sequencing using spatial 10x genomics spatial sequencing. So this is where you take like a tissue section of, say, a tumor or a certain tissue section that you're interested in, and then you can actually like I said, barcoding each of those individual cells and then sequencing each one. So you get some transcriptomic data that's has, you know, spatial aspect to it. Okay, and obviously this is what we're all here for today, is we also have a GridION, we have MinION, MK1C, we have Flongle. So we have a whole range of Nanopore devices that people, again, can book and use. We've got this set up in our iLab system just recently, so people can come in, even if they want to do their own library prep, they're welcome to come in and use the devices. Although obviously, Annabelle and I have been using Nanopore sequencing for a long time. I think I've been using it since 2016. So we're very experienced users of Nanopore. So we also obviously offer that as a service. So you can just hand us your DNA and we can sequence it for you. And that can, yeah, take some of the troubles. We can do the troubleshooting for you if there's some issues. Yeah, and so as I think Warren mentioned, you can basically sequence any DNA or RNA on this platform, direct RNA. It's real time. So this is a great thing too, where you can actually use it in the classroom, sequence with your students and show them the data in real time, like that their sequencing is working. Or if you have some clinical application where you need some data very fast, like for COVID sequencing, that's sometimes very time critical stuff. So yeah, it's real time data delivery. And Annabelle will speak to, we've been doing some adaptive sampling as well. So that's very exciting. So I'm going to hand off to Annabelle and she will be talking about all of the cool projects that we've been doing here and what you can do with Nanopore.
Kia ora. So yes, I just wanted really to pick up on some of the main use cases that we have here, just to highlight some of the things that people in Auckland are doing with Nanopore sequencing. And some of the questions that we get asked around how to scope these projects out or what to expect from them. So just really picking up our main use cases, but as Warren has said, there is a real diversity of things that you can do with this technology. So in-house, we have, as Nikki said, the GridION, we have MinIONs, and we have the Mark1C, and all of them use this same flow cell that's pictured here, this MinION flow cell. They just have either GPU or not, and sometimes you can use with the GridION up to 5 flow cells at a time, but the same kind of functional unit for all of these devices. And I'm going to talk through how we do these experiments, and because you monitor them in real time, you know how things are working and you know how things are progressing, and you get information back as you're sequencing. And this is all reported on the Minnow software, the kind of dashboard that interfaces with the technology. And these are really useful diagnostics, and I just want to take a little bit of time to explain some of the key ones that we use, because I'll use that in subsequent slides to talk about particular experiments. So at the top is the kind of channels panel, and it's showing you the 512 channels as they're arrayed on the flow cell. And this is quite late on in a run, this snapshot. So when you start, you hope to see lots of bright green, and that's great. These are channels that are actively sequencing. And over time, there's some kind of degradation in the pore activity. And so more turn to blue. The other way to map this same information is to do it what we call a duty time plot, which is this one here in the middle. And that's showing basically the amount of sequencing you're getting in time intervals across the course of a run. And this flow cell here is run for 18 hours and generating data in a proportion to that bright green part of the signal. And you also know properties of your library, so your read length distribution as you go. And this was just what shown one here, where the The N50, so half of the reads are 7.4 KB or greater in this particular library. And getting this information out right from the start of your experiment can be really, really useful. Sorry. Yes. So one of the questions we get very often is, what kit should I use to sequence? And there are largely the field kit, the rapid kit, or ligation sequencing, but also lots of different variants of that with different chemistries. I could give you answers right now, but it's quite likely those answers will be out of date very quickly. And so the thing that I really wanted to highlight here is that the Nanopore community website is a very useful place to go to find protocol information, and to also ask questions of the community if you want. It's very active. People give a lot of feedback. We're also here. You can ask questions of us and we can direct you as best we like. But there is a lot of information out there. And if you're new to this, it can be a little bit overwhelming, but panic not, because there will be an optimal solution for your project. One of the most common projects that we've done to date has involved sequencing whole genomes of bacterial isolates and generating really high quality, complete and contiguous assemblies. And this was one of the really initial selling points of this technology, is that you could get super quality assemblies because of enabled by the long reads. You can also multiplex, and so it becomes quite cost effective. And the accuracy is improving so much that you may no longer need to generate Illumina data to try and polish up the quality of the accuracy of your assembly. As Warren has said, we can look directly at base modifications, and we can support you with that at Auckland Genomics, and we can help you with the bioinformatics if that's something that you would need. We often get questions about how much sequencing should I try to get for my assembly. And in Illumina type experiments, that's often quite a straightforward answer. And I really just want to flag here that it's not just sequence quantity, but also the length distribution of those sequences that has some impact on the outcome. And this is just one, it's a yeast genome assembly to kind of illustrate that. And basically the number of contigs when you have a library of short reads, so the 1 to 15 KB are the shortest segment, they're all at 20x coverage. Coverage is not an issue in the comparisons. And you get many more contigs. Your genome is much more fragmented if you're only putting in short reads compared to if you can put in a section of long reads. And so you'll often not get a straight answer from us on how much sequencing depth you need to get the optimal assembly, because it's a more complicated, more nuanced question, I think, than a lot of us are used to with other technologies. As well as doing microbial genome assemblies, we've had projects doing eukaryotic ones. And actually, this is something that I started in 2019 with my research hat on. And I've talked about this a lot in different New Zealand forums, but I like to use this tweet from Nanopore, which talks about how the accuracy has improved in recent years. And so this is the plots are for a 2-year period. They go from albacore to Guppy 4. We're now on Guppy 6, and that improvement in accuracy has come on further still. And I like to talk about this because we live these improvements. They kept releasing new versions of Guppy or new chemistries, and it made a material improvement to our assembly. So I was working on the HeHe, and we've recalled base data and assembled that probably 4 times in the course of a 2-year COVID disrupted project. And I guess I also want to say, if you have experiences with this technology from a few years back, and maybe the performance of the product or the accuracy were an issue for you, you should look again, because it's really just quite incredible how much the progression has been. And it's a really, really exciting sequencing technology to use, and it enables a lot of the research that we can do. And this is my final HeHe assembly. So I'm just comparing, we generated 2, a male first, and we then learned from all our mistakes and generated a female too, and they're very highly contiguous and really nice quality genomes. As I say, read properties matter quite a lot. So the male HeHe, we initially didn't do any size selection because we were really limited for sample quantity. And then once we got a decent amount of coverage, we added in some longer reads. And just 10% of those longer reads really made a dramatic improvement to the assembly quality. So we ended up with about half the total number of contigs just by boosting that long read component. Another question is, well, what yield am I going to get out of my flow cell? How much data can I expect? And actually, it slightly depends on the properties of your sample. So if you have a great sample that performs very well and can maintain performance across the time of the run, then I would say you're guaranteed to get 10 to 15 gigs of data, and quite possibly you could get more. We wouldn't guarantee that, but Nanopore have reported actually 50 gigs from a minion flow cell in the most optimal conditions, and 30 gigs is not unusual over a 3-day run period. But a lot of the samples that we do are a bit more problematic. They do block pores, and there your actual the amount of functional sequencing capacity of your unit can decrease quite quickly, and then you will get less out in the end. And sometimes that's intrinsic properties of the sample, and other times it's actually something a little bit different. And I have a recent example from a project that Dr. Libby Liggins at Massey University has been involved with in collaboration with Tifano and Akamu, working on the sea urchin or Kina. And this is just 2 different extraction methods from the same tissue. The one on the left is a high molecular weight extraction protocol. You've got a read N50 of about 30 kb. The same tissue extracted with a spin column, which is not optimal for this kind of tissue. It becomes quite degraded. We've got a read N50 of about 7 kb from the exact same starting material. Swings and roundabouts though, because this long extraction actually blocks the pores a lot more. So we struggle to get really good yield out from these long molecules, whereas these fragmented smaller molecules actually are less problematic for the sequencing. So this project will end up with a mixture of these 2 types of libraries, and actually that should come together to produce a very nice quality assembly. We've recently done some adaptive sampling, so this in silico enrichment in this particular case of genomic targets. So this is work that Peter Tsai and Paula Shields in Professor Chris Prince group were leading. And what we found was that also what you see characteristically is this peak of very small reads at the beginning of the read distribution, and those are the rejected reads. And then the ones that the device chooses to retain, those are longer, and those are the ones that you're interested in. And so we're enriching for about 1.2% of the genome and recovering just the regions that we're interested in, which means to achieve it's much more cost effective essentially, and a really nice way to just focus your sequencing effort on what you're interested in. Amplicon sequencing is another really big application, and the one that people are most familiar with probably is the way that long amplicons were used for SARS-CoV-2 genome sequencing. And this is a 1200 base per amplicon panel called the Midnight panel that was developed by Nikki sitting next to me and Olin Seilander, and has been really widely used. So there's a large number of the genomes that are in the GISAID database were generated with nanopore sequencing. The real-time aspect of this is really powerful, and also the ability to come up with an assay that is very robust and actually could be tweaked when necessary, when new variants evolved in a really effective way. And so if you have amplicon sequencing projects, again, this is something that this machine can handle very effectively. Warren talked about methylation, so I won't really dwell on this, but we looked, for example, at the methylation profiles in male and females, and you can see the level of methylation is very correlated for the same genomic window between the 2 samples, for example. And that's something that there's a big evolution in the pipelines available to study this, and it's a really exciting development of the technology. So more waiting room people. I'm rubbish with the mouse.
Sorry. Great.
Anything to change? It's also a really, really good tool for outreach. And so one of the things you can take it into schools, you can take it out to community groups. It's really neat in kind of an environmental DNA setting, for example. And so it's one thing to bear in mind, and we can support you in doing that. And taking out into the field sometimes is a really useful feature. And we're also, just to say that we're very interested in providing training. So I've actually trained a number of people in Auckland over the years in how to use this technology. If you want to get started, you don't know how, please do come to us. And we also hope to organise some in-person workshops later in the year. And so if you have particular training needs, please do get in touch, and we'd love to support you in that. And I think that's all that I was going to say. So thank you for listening.
We can now sort of hand over to Mike, I think, and I will stop.
It's going to Warren for a quick.
Oh, sorry.
Yeah, he's going to do a quick update That was done at Nanopore community meetings, some other things, and then I'll move on to myself.
Yeah, Mike's follows on from mine quite nicely. He dives into some in more details.
Okay. Good job.
Um,
Over to you.
Yep, thank you very much. My next presentation is an update. So where the platform is with respect to recent developments that were announced at our Nanopore community meeting in December. And then Mike's going to take some of those developments and give you a more recent update in the few months since December. There have actually been more improvements that have been released. So he'll explain those because it's really important to understand what's happening as our technology develops, because that affects your decision making for the work you're doing immediately and in the near term. So I'll start this presentation with a slide of the real Nanopore family of devices. So the one I showed earlier was for instructive purposes only. And you might notice that there are two new devices shown here. It's the PromethION 2 Solo. They're in development, but they're very soon to be released. So these devices will run the PromethION flow cells, but instead of having slots for 24 or 48 of them, there's a slot for 2 of them. So it's a small scale PromethION, which is fantastic. We've been waiting for this to be developed and released for a while now. So the other fantastic aspect of this is the very low cost of entry for these sequences. So the PromethION 24 was a fairly expensive box in our terms compared to other high-end sequences. They were a fraction, but we've got a really cost-effective tool in the PromethION 2. The first of, actually, I'll talk about those in more detail in a minute, but the pricing you can see here, I'll just explain. There's no capital cost. So the prices you see here are our starter pack prices. And those prices are the cost of the flow cells and kits that come with the starter pack. The starter pack, you can think of it as 12 months of consumable usage. And so it provides a starting point at our standard pricing, and we include the device free of charge. We actually retain ownership of it. And that's an advantage because it means as we develop it, we can swap in and swap out boxes. If there's a problem a hardware issue that can't be fixed on site, we don't have to send someone in. We just send you a new one and you send the old one back to us. So that has some advantages, but it ideally means you can get started with Nanopore with project money because you're just buying consumables, or you can tell your funding body that it's a device purchase, inverted commas again, and you get a whole lot of flow cells to start get the project moving. So it's got some nice advantages there. So the PromethION 2 is shown on screen here. So the PromethION 2 it's only very small. It's about this big, incredibly small. We are launching the P2 Solo first. This will be available before the middle of the year. We'll be starting to ship, and then we'll be catching up on all the back orders. P2 Solo has no built-in compute, no GPU built in, which is why the price of it was only $10,400 and included 8 flow cells. So because it doesn't have built-in compute, there are 2 solutions to that. You can actually plug it, like the MinION, original MinION, you can plug it into your own compute facility. It needs to be GPU based, or you can plug it into the GridION and use the GPU in the GridION, which a lot of core facilities are planning on doing, because it gives you PromethION capability at a very low cost compared to the PromethION 24 option. And then we've got the P2 sort of standard system, which will have a built-in GPU. It's going to be available before the end of the year, and it's still in development, as I said. But you can still register your interest on our website, and you can actually get quotes for these, And you can also actually place orders for them. We don't mind taking orders for products we can't send you, but we're going to send you them in the near future as soon as they're released and they're available. So have no fear. We won't charge you until you actually receive it. Another development around the PromethION platform is this extended runtime. And we're calling these marathon flow cells. And they're going to be rolled out in the next month or two and replace the standard flow cell that we have. Basically, they've got deeper wells so that there's more reagents in them. And if those reagents become limiting during the run of the standard flow cell, compared to that, you're going to generate 10 to 20% more data. Now, it doesn't affect reduction of output caused by blocking or poor degradation, but it will improve runs where it's the reagents that become limiting, because much like a battery, as the ions are flowing through the pore, the difference starts to decrease across that membrane, and eventually the difference is significant enough to end the sequencing run. So we're improving the PromethION flow cells. Now, Annabelle touched on sequencing accuracy, which has been, I guess, a key focus for us over the last few years, but it's not so important now, as I'll show you. So similar graph to what Nikki showed. Over the last 2 years, our sequencing accuracy has increased from about 92, 94% single read when I joined 3 years ago, and it's now over 97% just for algorithm development alone. And that's been really awesome, because it means you can just rebase call your old data. And we're going to still drive improvements in our base calling software I'll show you in a slide or two. And on the right-hand side here, you can see we've also initiated chemistry improvements. Our current state of the art is using Pore 9 with Kit 10. So the kit is the chemistry, the adapter, and other chemical changes. The pore is the pore design, which involves mutated variations, and we find ones that work better. Pore 10 has been sort of in early release for quite a number of years, and we've been continually working on it to get more accuracy and higher output, and we're there. So Pore 10 Kit 12 is the very latest improvement in that, and Mike will be talking more about the next one that's coming after that. But with Pore 10 Kit 12, we can achieve accuracies over Q20. And that's why we're calling it Q20 plus chemistry, because it describes where we are with our level of accuracy. And there's an example here of what that means in practical sequencing terms. So on the left-hand side here is a Zymo mock community running our old Pore 9 Kit 10. And that was the state of the art 6 months ago, to say, but it's regarded as the old legacy chemistry. We're supplying it for the next year or so or more, because there's projects that are using it, and we're not going to cut them off. But we're really encouraging transition to the new pore chemistry. So you can see with the Zymo mock community, at about 50-fold coverage, you're looking at around Q45 for consensus accuracy. With the new Pore 10 Kit 12, the same level of, well, actually, we achieve approaching Q50 accuracy with less than half that level of coverage. So less than 25-fold coverage gets us the same or better accuracy. And you should see this across all genomic sample types. Another improvement we've made recently is for plant sequencing. Now, plant DNA has a range of undefined modifications that we call as errors in the software. And previously you were able to train your own, develop your own data sets and train our algorithms to remove these from the sequencing analysis, but we've finally done that. So we've incorporated a maze data set that has no modifications on it, and we can train our base callers to identify those sequences. And so we now offer the same level of accuracy with plants as we did with microbes and with human DNA, which is fantastic. And there's some icing on this cupcake, and that's called duplex sequencing. Now, duplex sequencing occurs when the secondary strand follows the primary strand through the pore. And in a typical sequencing run, that occurs in sort of 3 to 5% of the time. What we're enabling now is the consensus calling of that duplex run, those duplex reads. So both strands are consensus called across that. And that enables an accuracy for that strand of approaching Q30. And in fact, Mike might, in his talk, might touch on the fact we're now over Q30 for some recent additional improvements. And the other thing that we've enabled is the amount of duplex reads that occur in a run. So tweaking our chemistry, we've increased the percentage of duplex reads that occur to easily 10 to 20%. And our goal is 30, 40% at least. And so we'll be continually tweaking the chemistry behind the scenes. And so you'll be able to get these higher level accuracy duplex reads available in your data set to analyze either separately, so you can have an incredibly accurate component data set as a segment of your total sequencing run, or you can combine it with the regular simplex reads. So if you were to run a human sample on a PromethION flow cell, what do you get? Well, taking a standard human genomic sample on a PromethION flow cell with our highest throughput chemistry, you generate about 150 to 180 gigabases of data, which is about 60-fold coverage. Using POR 10 kit 12, the accuracy for SNPs on that data set at 60-fold coverage is 99.9% precision and recall. Indels are 89%, and we're now measuring indels across the whole genome, not on filtered regions of the genome. Most other genomics companies choose the regions that they determine the accuracy of indels. And so it's a little bit misleading, and we were guilty of that too, no longer. Our indel accuracy is whole genome, unfiltered. That's what you can expect. Structural variants are similar, 96% precision and recall. And as it's strongly shown in the published literature, long-read sequencing can identify 4 to 5-fold the level of structural variance compared with short-read data in the human genome. So that's incredibly important. And the biological implications of structural variance are slowly being teased out. And there's a lot of work that's going to be done over coming years in that area. CpG methylation correlation with bisulfite sequencing is over 98%. So I showed you earlier, I think 93%, it's over 98% now. POR 10 actually is better at determining methylation status than POR 9. So we get some bonus improvements. And sticking on methylation for a minute here, we're about to, we've developed a new tool called Remora for calling methylation. The previous one that was sort of best in class was Megalodon, and it was really GPU intensive. You couldn't use it on our systems. You actually had to download the data, and it took days to analyze a large data set with Megalodon. Very accurate, but it took a long time. This new model, and I'm not sure of how it differs from other models, but it's very GPU light. In fact, it's so GPU light, we're including it with a pending release of our Guppy software for analysis, so that you'll get the nucleotide calling and the methylation calling at the same time in real time by the end of a sequencing run, which I think is really amazing. Mike, hopefully we'll talk about the new base caller that's coming. It's not here yet, but we've even got an improvement on Guppy. Anyway, back to the right-hand side here, there's also an improvement in bisulfite analysis. You can, as I mentioned, you can see the increase in the accuracy is about 1%. Now the correlation is 0.985. So this actually is awesome. It's becoming the gold standard of methylation. An interesting case study here using adaptive sampling for methylation analysis. It's basically reduced representation methylation sequencing that a lot of people do with bisulfite. You can now do that without any enzymatic or any wet lab modifications. You can see on the right-hand side here, using traditional bisulfite sequencing, it's a couple of days of enzymatic and sample processing compared with none with Nanopore. It's just your standard library prep and download the data set into the sequencer. And just to give you a comparative aspect to that, looking at bisulfite sequencing 30-fold coverage, you can identify about 3.7 million CPG sites. With adaptive sampling at only 20-fold coverage, you can identify 6.3 million CPG sites. And the cost for this is incredibly, it's really good. So a flow cell for around $500, library prep for $100, you get a lot of data, a lot of accurate data. And in this particular case, I forgot to mention they identified 18,000 regions for analysis with the adaptive sampling approach. So very powerful, and I think we can replace reduced representation by sulfite sequencing with this as groups give it a go and see what it can do for them. Another important aspect I don't want to skip over is our ultra long read sequencing kit. This was released about a year ago, so it's not brand new, but it's incredibly useful. It really helps us with genome assemblies, resolving gaps in reference genomes, spanning centromeres, telomeres, and complex structural variants. And also it simplifies phasing and improves phasing of SNPs, phasing of indels, phasing of methylation. On the left-hand side here is a run from the Garvan early last year, blood on a PromethION flow cell, 88 gigabases, which is pretty good, with an N50 of 102 kb. And you can see the tail of ultra long reads is substantial, going right out into the hundreds of kbs, which makes so much, which really benefits assembly and structural variant analysis, etc. Just to drive this home a little bit further, if you're not convinced, on this graph here on the left-hand side, it shows across the bottom here different library sizes: 11, 17, 28, and 30 kbs. And on the axis here, you can see phase block length. So a 10, 15 kb library will generate a phase block length of 200 or 300 kb. If you double that library size to 30, 35 kb, the phase block length increases 4 to 5 times. So it's not linear. So that doubling of library size really has a huge impact on assembly and phasing. And on the right-hand side here, looking at 30-fold ultra-long read data, this increases the phase block length to over 10 megabases, which in many cases is chromosome level assembly and phasing, which is just amazing and incredibly useful. Touching on the sample preparation kits, Mike might be going into this a little bit more detail, but there's one point I just want to make here, in that our rapid and field kit is currently nearing the end of a new round of development, and we will be releasing new version in coming months. And one of the key aspects of this version is the amount of DNA required drops from about 400 nanograms for the old kit down to 50 to 100 nanograms. And so this is going to be really useful where there's limiting sample availability. And I'm thinking possibly environmental samples or culture samples where there's not a lot of DNA that you can get out of poorly growing strains. I'll touch on short fragment mode. Mike's hopefully going to cover this in a little bit more detail, but as I mentioned in the beginning, we can go down to 20 base pairs. we've basically disabled the 200 base pair cutoff filter that was in Minnow, and we've built in some slide selectors, so you can choose the size of the fragments that you look at, but we can go down to about 20 base pairs. And this is going to be really useful in liquid biopsy, and I think in environmental analysis as well, because 100 base pair sequence is still really useful for identification purposes of microbes and other species DNA that might be in that environmental sample. And to just give an idea of, if you run a sample on a PromethION flow cell that's around 200 base pairs in length, you get over 250 million native reads generated. So the output is incredibly high. As I get to the end of this presentation, this is the newly released PCR cDNA kit. If you've done any transcript analysis with our previous kit, this offers substantial improvements because we're using DNA sequencing in this case of your reverse transcribed RNA. We've been able to take advantage of the improvements in DNA sequencing, and one of those is the fuel fix adapter. I won't explain the details of that now, but it means you get a lot more reads from a sequencing run. So you get well over 100 million reads from a PromethION and well over 15 million reads from a MinION. Those figures there are best in class. They're what we can achieve internally sometimes, but you definitely get up towards those in the real world. And these are all full-length transcripts. So it's incredibly useful data. We've actually modified the assay so that it removes internal priming artifacts. These used to make up 10 to 20% of your data set and will be filtered out. They're no longer generated, and therefore the increased output plus no artifacts means you get so much more useful data. The new assay actually primes off the tail, off the very end of the poly A tail. So you get the full length poly A, and there's a nice little tool in GitHub called Tail Finder, which enables you to estimate the length of that poly A tail. We have got UMIs incorporated into this assay for future analysis, for future quantitation, and barcoding kit is 24 barcoding kit is going to be coming out in the next month or so at the latest. This kit is the cornerstone of our new single cell and spatial assay, as Annabelle mentioned. It's been possible for quite a long time now to follow some published protocols for full-length transcription analysis from single cells or spatial, utilizing the Visium or the 10x system. However, there were some limitations because our accuracy wasn't so high. The UMIs weren't as accurately called, and therefore the recommended workflows involve splitting the sample and running half the sample on short read so that you could generate accurate estimates of the UMIs, and you would use that to help identify the UMIs for the long read sequencing. With our improved accuracy and improved data output, that's no longer necessary. We've got a protocol on our community site now. The one thing that's missing is demultiplexing of the 10x barcodes. We're working on that feverishly, and within the next 1 or 2 months, that's going to be released. And so this will be a fully enabled protocol that's Nanopore supported. And so if you're interested in doing any, I guess, deeper dive into the biology of single cell or spatial genomics, this is the way to do it. 3' counting is going to give you an estimate at best. And there are many applications, for example, neuroscience that has shown that isoforms are very important at the single cell level. And so we'll be enabling that. I'm just going to finish my talk here with a mention of one of our initiatives that was started last year, and that is called org1 or org.1. And this is targeting critically endangered species worldwide. And it provides reagents and flow cells free of charge to have sequencing done on a species that you nominate that's on the critically endangered red list. If you've got, I believe there's a dolphin species down from Otago Uni that's been sequenced by this program, and we've got a range of others that are in process. You just go to our website, have a look at the organisms there, have a look if it's been done or not, and you can register your interest, and then we'll be in contact with you to discuss the details of where you can get this sequencing done. It encompasses both MinION and PromethION flow cells, and we'll work out the best way and where you can get that sequencing done. Several species as examples that have been done recently, the hurricane palm, the Mauritius olive white-eye, and a Boger's skink. So it's to really help researchers get some valuable genomic information on species that they're interested in that have environmental importance. I withdrew a slide from here that actually shows that we're going to be broadening this into environmental DNA as well as organisms. So have a jump on the website if that's of interest to you, and see if we can contribute to some vital research. So that's pretty much the end of my talk, and thank you for listening.
Hi, thanks, Warren.
If there's any immediate questions, I'm happy to take them at this point.
I see that there's one in the chat.
There's a lot in the chat, yeah.
Oh, there. I'm just sort of.
That's all right.
Yeah, maybe Warren, in the effort of time, maybe you could answer the questions in the chat.
Okay. Yeah. While Mike's going on, I'll run through those, and
then everyone could take a look. That'd be great.
Good idea. Good idea.
Okay. Looks like Mike is setting up his stuff, and I will stop recording because this is all so brand new. Resume the recording just in case. So we also, a few people have asked us, we set up some time after this to do little sort of one-on-one discussions with people. So we have a few people who've asked us to meet after the session, and we'll go through some, if you have something, how you might want to use nanopore sequencing in your workflow or your experiment, your research. We're going to do a few one-on-one trainings, and obviously if you didn't sign up for that, we're always here. We're happy. Annabelle and I are always happy to chat with anyone about their projects. Afterward, I think we've got Mike and Warren to stick around too. So yeah. Anyway, I've been amazed. Every time I take a break, a month or two, and don't keep updated, something new happens. And so it's just such a rapid pace of progression, and it's amazing how fast the quality is improved, and just all the different ways you can sequence DNA and RNA. So it's really exciting.
Just thank you for joining us today. Please get in touch with any questions, and huge thanks to Mike and to Warren for sharing their expertise and for their continued support.
Yeah, absolutely.
Thanks, everyone. Yeah, I put my email address in response to a question in the chat box. So anybody who wants to contact me directly about any aspect that they'd like some more detailed information on, after speaking with Nikki and Annabelle first, obviously, I'm more than happy to. And look forward to catching up with some of you in New Zealand later this year.
Yeah, we'll be running some workshops. We have some workshops planned for later this year. So hopefully some hands-on training and a little bit more, really how to get in there and get your sample sequenced.
Queenstown Research Week.
Yeah. Okay, with that, I think we'll just close off the meeting, and
we'll take 5 minutes for those who are coming back, and then we'll join on the other Zoom
call.
Perfect.
Okay.
See you there.
Thank you.
Okay, bye.
Yeah.